Thursday, so we were supposed to have it today, but we moved it back one day, so hopefully you saw that announcement. It's going to be chapters 5 through 8. I have to actually stay home tomorrow because my babysitter is sick, so I'm the one who has to stay home with my kids, which my kid actually got the babysitter sick. I'm not sick, so I have to take care of all this, so I don't think I'll be able to have a live review meeting with you guys, which sucks, but I think I have a recording from a previous semester, so as long as you don't mind other stuff and just ignore that there's other people maybe, I'll see what I can come up with, but I'm definitely going to post something. So most people's questions are about chapter 8, and hopefully we'll clear up some of those questions today. So I apologize for that. It's going to be pretty much impossible for me to go live at home with my 3-year-old because he's insane. So I apologize, but hopefully in the future that won't happen. So I will post something for you, like some kind of recording, just kind of going over in general what we've talked about. If you guys have specific questions, of course, you can ask me. So again, to recap, Thursday, we're just going to take our exam, chapters 5 through 8. I think it's worth 75 points. It's going to be just like the last exam, where it's a mixture of matching and multiple choice. And remember, if you look at the outline, if you're ever asked to give an example of something, you should be able to give an example, although I don't think that came up that often in this chapter. We talked about soap and how you grow the microbes and a bunch of other random stuff. So if you look at the outlines that are in your notes, your class notes, and do the worksheets, I think you will be plenty prepared for the exam. The next set of notes I just sent to the printer because I'm a procrastinator. I was like, could you have them done by Thursday? And I'm sure they're going to be like, no. So anyway, they'll probably be here by Tuesday. So I'll give you another booklet like I did. If you're someone who likes to look at the notes ahead of time, I can just post the electronic file online. But you don't have to print it if you don't want to. You can just wait until you get your copy on campus. So that'll be coming also. I totally don't even remember where we left off. It feels like it was 8 years ago. We talked about amino acids, central dogma, right? I think that's where we left off.
Yeah.
That's usually a good stopping point. Does anyone have any questions before I get started about class or anything? Everything okay? We're all right? Okay. So I did post the practice quiz. I got that up last week. So that's good to help you prepare for the exam. I haven't given a lot of homework this semester just because it's been so crazy. And I still have to post whatever extra credit opportunities that I come up with. And we are going to have lab today, so we're going to do lab number 3, and it's pretty quick. So just don't forget to do your quiz on lab number 3 if you haven't. And I have been posting PowerPoint slides for labs. I didn't print them because sometimes they're better in color. And then I've been posting videos too that support that information. So that might be helpful later when you study for your final for the lab because it is cumulative. All right, I'm glad to see you guys. I'm like, whew, man, it's crazy. I don't know if we should be here. Do you ever feel that way? Maybe we should be maybe we should all be at home chilling, getting paid by the government to relax until COVID goes away. Wouldn't that be nice? I mean, I'm not someone who's just give me money, but at the same time, I'm like, where's my stimulus? You guys are just talking about other stuff. Anyway, I'm glad you guys are doing okay and staying relatively healthy. So we'll get through this. Okay. So genetics is what we left off talking about. So we talked about DNA versus RNA, and we'll use those little handouts that I gave you as a review at the end. And I'll post the answer key, and I will post an additional genetics problem and let you know what I mean by that, what would be on the exam that you have to know how to do. And in fact, I think if you've taken the quiz already, you've seen what I'm talking about. Where if you see a strand of DNA, you have to make the opposite strand, and then you have to be able to make the RNA sequence, and then you have to be able to make the protein amino acid sequence. So there's a chart you use that's the genetic code, and that's on one of the handouts I gave you. And you don't memorize that at all. You will actually just use that chart to help you get your answers. And so you'll see that when I post the other practice problem with the answer key, so you'll be able to check yourself. All right, so we left off talking about central dogma. So this is basically how we believe living organisms deal with their genetic information. This is the flow of genetic information. And so essentially, genetic information is written in a 4-letter code that is the basis. So that's the A, T, C, and G that are on the DNA molecule. So your genes are made out of DNA, and genes encode for protein. But before you can turn them into protein, you have a little step in between where you actually convert them into RNA. And when you go from DNA to RNA, that's called transcription. And then you take that RNA and you turn it into an amino acid sequence, which is what you need to make a protein. And when you go from RNA to amino acid, that's called translation. So there's different enzymes that will run these processes. If you want to go copy your DNA, that's called replication. If you want to make an RNA, then that's called transcription. And if you're going to start to make a protein, that's called translation. And so there's different parts of the cell that are involved in this process. And this is how we think that the flow of genetic information goes. And the whole point of the genes is that they encode for proteins, which are what make up the traits. And another word for traits is characteristics. So everything about a living organism is encoded in their genes. And the genes each have unique sequence of DNA, of the 4 letters that make up DNA. All right, so if you're going to copy your DNA, that's called replication. And we mentioned last time that this takes a lot of energy to copy your DNA because it's a huge molecule. You need to copy your DNA anytime you're going to divide. So anytime a cell is going to do cell division, it has to replicate its DNA before it can fully divide. Because every cell of the organism has to have a full set of the genetic information unless they're sex cells. Sex cells like sperm and egg only have half of the genetic information, but all the other cells have to have a full set of however many chromosomes that organism has. So humans, we said, have 23 pairs of chromosomes, 1 from each parent. And then bacteria actually only have 1 chromosome, and that's because they're clones. They're clones of each other. They reproduce asexually, so they just make copies. So they just got their copy of their chromosome from the cell that they came from. So the process of replication is what they call kind of conservative, because basically each new strand is 1 part old strand and 1 part new. Essentially what you do is it's a double-stranded molecule. You open it up, and then you copy from each side, and that's how you end up with 2 identical copies. So the reason that this can be done by you can pretend to be an enzyme, essentially, is that there's complementary base pairing. A always pairs with T, and C always pairs with G. And so if you remember that, then you can make any complementary strand. So usually the strand that you start with is called the template strand, and then the opposite strand that you're going to generate is called the complementary strand. So this is a necessary step for cell division. All right, so this is very oversimplified the way I'm explaining it because there's a lot of different enzymes involved. And this is something that's just happening in your cells all the time, which is like pretty amazing. So remember, anytime you see an ACE, that's an enzyme. So helicase, its job is to unwind the helix. And so they call it a helix because it's 2 strands that are coiled on each other. So imagine a ladder that's been coiled. That's basically what a DNA molecule looks like. So helicase will come through and it'll unwind the helix. And then there's another enzyme that's called DNA polymerase. Polymerase means to elongate. So that enzyme is going to come through and it's just going to read the sequence, and every time it sees a base, it's going to add the complementary base to that growing chain. And because of the way the enzyme works, it actually does it in like 2 different directions. And really, it's actually doing both strands at the same time. It makes like this weird cross, like with the 2 strands, it makes like this weird square. It's kind of hard to imagine, but it twists on both of the strands so that it can do both, I guess, at the same time. It's like really weird. It's like a little machine that does it. And then we don't even get into it, but there are proofreading enzymes that'll come through, and if there's any wrong letter put down, they'll remove it, which is like pretty amazing, and that's how we prevent from making mutations. So the old strand and the new strand is in both of the strands, because remember, you open up the original, and then the red strand is the new one that's getting put down due to the complementary base pairing. So this is just enzymes that do this. You open up the double helix, you use DNA polymerase to copy both sides in different directions. One of the strands is going to get synthesized faster than the other, and that's called the leading strand. And the other one is going to be a little bit slower, and that's called the lagging strand. And just because of the way the enzymes work, the lagging strand is slower because they actually have to like glue the individual pieces back together. again, just the way the enzymes work. So this is showing it another way. You might notice that it says 5' to 3', and I usually don't mention it, but people will sometimes ask about what that means. It has to do with the directions that the sugars are facing on the DNA. And so if you remember, a DNA nucleotide has a deoxyribose sugar on it, and it looks like a pentagon, and that's because there's 5 carbons in that sugar. That's why it looks like a pentagon. And they'll label the sugars 1, 2, 3, 4, 5. So on one of the strands, carbon number 5 is the one that's up. And on the other strand, carbon number 3 is the one that's up. And so they're just oriented in different directions. And that's why they're called anti-parallel, because they're actually, the sugars are facing opposite ways. And that comes up, if you were really to take an in-depth molecular biology class, you would talk about the directionality of the enzyme and like how it has to read the 5' to 3' strand. So the 5' to 3' strand is the template strand. That's the one that you would use to make a complementary strand of DNA. So again, the lagging strand is slower because it has to be pieced together in fragments that have to be glued back together, and those are the Okazaki fragments. And then helicase, this point where the helix is opening up is called the replication fork. And helicase will just continue to unwind it, and then DNA polymerase will come by, and it'll just add complementary bases. So if it sees a G on the original, it's going to add a C. And if it sees a T, it's going to add an A, and so on. So you just have to remember that the T's and the A's are going to pair up, and the C's and the G's are going to pair up on a double-stranded DNA molecule. Okay, so that's replication. It's when you copy your DNA, you're synthesizing a new DNA molecule. So 1 becomes 2, and you usually do that for cell division. Okay, the next step then, if you're actually going to express the gene, like you want to do something with it, then you have to do that little in-between step, which is called transcription. So transcription is where you're going to open up the original DNA, and now a different enzyme that's called RNA polymerase is going to put down RNA nucleotides instead. So the difference was that RNA nucleotides have ribose as their sugar, and instead of T's, which is thymine, they have U's, which is uracil. And this is the part that confuses people when they're going through their process. For whatever reason, the way I think about it is that when the ribosome goes to make a protein, it just doesn't understand thymines. So it needs uracils instead. And so you'll notice that the genetic code in the table, there are no T's in the code. They're all U's instead of T's, because that switch is made when you go from DNA to RNA. So transcription is basically converting the message that was on the double-stranded DNA into a message on single-stranded RNA. So the enzyme RNA polymerase, it both unwinds the helix and it adds the new bases. So it actually does both things at the same time. All right, so this enzyme is called RNA polymerase. It makes the RNA longer. Transcription starts at a special site on the DNA that's called a promoter. That will promote transcription. That's where you know to begin the sequence. And then there's also a stop signal that's called the terminator. And then once the RNA and the RNA polymerase is released, the double helix will go back together because the original DNA will always go back to its form. And now you have a new molecule that's called messenger RNA. And messenger RNA, to me, it sends the message of the original DNA to the ribosome now. So I think of it as like your DNA would be like your original copy of something you have. Let's say you have an original copy of you have your driver's license, right, in your hand. But if you needed a copy of it for some reason, and maybe you needed an official copy or something like that, you needed notarized, that would be the RNA, would be the official copy. It's the same information, but it's a copy of that information. It's not the actual original DNA molecule. And they probably have to protect it that way because the DNA is critical for the cell to stay alive. So you don't want to lose any of that information or have it be damaged in some way. All right, so the promoter region on DNA lets RNA polymerase, which is that little blob-shaped thing, it lets it know where to start, right? Because it's a whole sequence of letters. It's where do I start? So there's a start sign, a start line, if you think about it as a race. And so RNA polymerase comes through, and it'll start opening up the helix, and then the RNA nucleotides get added, and it's just complementary base pairing. But the thing that changes is that if there's an A on the DNA, there's not going to be a T put down, there's going to be a U instead. Because when you're trying to make an RNA, there are no T's on the RNA molecule. So anywhere that there was an A on the original DNA, you're going to base pair it with a U instead. And so that process keeps going, which is called elongation, as you keep making a long chain of messenger RNA, which is also called the transcript, because this process is called transcription. And then eventually it's going to read a termination sequence, and then it's like, bye, and then this comes back together, and now you have a transcript. So my understanding is that basically, in a simplified way, you'd have one transcript for every protein that you want to make. So if you want to make 10 proteins, you're going to have 10 of those transcripts. You've probably seen the term transcript in real life, right? Like if you ever looked at a YouTube video and they ask you if you want the transcript and it's like the written version of it, it's the same information, it's just written instead of visual. And so it's the same idea with transcription. Whatever was on DNA is now on RNA. And the reason that works is, again, it's just because of complementary base pairing. Because anytime you have those pairs, you can make the opposite match with it. All right, so then the last step is translation. So you're going to translate that messenger RNA sequence into a protein. And we mentioned that proteins have like a primary, secondary, quaternary, or tertiary quaternary structure. We just stop at the primary structure, which is simply the amino acid sequence. So proteins are made of amino acids, and amino acids are connected by peptide bonds, and so they'll also call it a polypeptide. Okay, so polypeptide is what is getting generated after translation. So what's going to happen is that the ribosomes are going to take the messenger RNA, and then with the help of ribosomal RNA and transfer RNA, they're going to bring in the correct amino acids by reading the genetic code. So that table that you would always have reference to, you wouldn't memorize it, it has all the codes, and there's 64 possible codes. And the genetic code is triplet, so every 3 letters on DNA is going to be one codon on RNA, and that's going to be equivalent to 1 amino acid on your peptide. So you read every 3 letters to make the coding. So there's only 20 amino acids. It's kind of amazing if you think about it. All of life is only made out of 20 amino acids, like only 20 proteins. But it's the way that they come together that gives them their functionality, like in their 3D structure. And mathematically, because there's 4 letters, A, U, C, and G, there's 64 possible codons to make 20 amino acids. So that's what they mean if you see that the code is degenerate. It means that there are fewer amino acids than there are codons. The advantage to that, though, is that you might avoid mutations or mistakes because you have more than one chance to get it right. And you'll see that when I show you the table, that basically some amino acids have more than one code that could mean that amino acid. And so even if you make a mistake, you might still get the right answer, basically. All right, so this is the genetic code, and as I mentioned, 3 letters on DNA is called a triplet, and then 3 letters on RNA is called a codon, and that's what's in this table. This table is full of the 64 codons, and then 1 codon is equal to an amino acid. And then many amino acids equals a polypeptide, and then those fold together in other ways to make a protein. So a few things to note is that there is a universal start codon that pretty much all polypeptides will begin with, and it's this guy here, which is AUG, and that's methionine. And I like to mention that because if you do a problem and it doesn't start with methionine, then you've done something incorrect. Like you might have used the wrong strand. They should all start with methionine, this met. Amino acids have a 3-letter abbreviation that is typically the first part of the word. And then it also has a 1-letter abbreviation, so you might see both ways. And I actually don't remember the 1-letter abbreviations as well. But I just know some of these I forget, like THR, for example, is threonine. and LYS is lysine. You don't have to know that, like, because I know some of you might not have had biochemistry yet. You just have to put down the 3 letters. The 3 letters is what shows me that you picked that amino acid. So the 3 letters with the A, U, C, and G came from the RNA. And that came from the DNA, which we'll go through how that is. So there's a first letter, second letter, and third. But this, I like this table because it already has all the codons in it. So you don't have to use that little tracing mechanism to find it. But what I wanted to point out about the code being degenerate is using serine as an example. So serine is an amino acid that has at least 4 codons and then 2 more, so it's got at least 6 codons for serine. So what that means is that if you made a mistake, you might still get serine because there's 6 possible codons that still mean serine, and as long as you put a serine there, it's probably not going to change the function of the protein. The other thing to note is that there are some stop codons. So there's 3 stop codons, and if you've done whatever thing you're working on correctly, there should be a stop. There should always be a stop at the end. And then that means stop, like that's literally the end of the protein. Like that's where the ribosome would stop translating. Okay, so that's the genetic code. It's a 3-letter code that came from the original genetic information. You transcribe it into RNA, and then that code is going to be translated into amino acid. Okay, so that's why there's no T's in this chart, because this is not DNA, it's now RNA. RNA is where the codes come from to make the amino acids. All right, so the ribosome is what's going to actually produce the protein. So this is the site of protein synthesis or translation. So basically, there's going to be a start codon, and that's where the subunits of the ribosome are going to come together, and they kind of like clamp down over it like this. Like if this was the RNA, it's like in the middle there. So it's going to start at that start codon, and then the ribosomal RNA that's attached to the ribosome is going to help stabilize that messenger RNA, so it's like in there. And then transfer RNAs are going to come in, and they're going to read the sequence, and if they recognize it through complementary base pairing, they're going to leave that amino acid, and then they're going to exit the ribosome. Then the ribosome is going to bond those amino acids together. It moves over to the next 3 in the sequence. Transfer RNAs are just flying in, seeing which one fits. Whichever one matches will leave its amino acid. It exits the ribosome makes the bond, it moves over by 3, and it just keeps doing that like crazy fast, like nanoseconds. Okay, so it'll just zip out this polypeptide. And then once it reaches the stop codon, that's the end of the polypeptide. So every 3 letters on the RNA is going to be 1 amino acid. And when it's 3 letters on RNA, that's called a codon. And it's the codons that the ribosome is reading. Like that's where the genetic code comes from. Okay, so one little side note. If you remember that we said bacteria don't have a nucleus. And so because of that, they can do transcription and translation in one step. So as soon as they start making RNA, they can immediately start translating it. But eukaryotic organisms are a little bit different. Since they have a nucleus, they actually have to do it in 2 separate steps. So they'll do transcription in the nucleus, and then the RNA will snake out of the pores, and then they do translation out in the cytoplasm. So because of that nucleus, they have to do it in 2 steps because it's 2 different compartments. And then eukaryotic organisms are also more strict at regulating their genetic material. So they do more proofreading before they actually send it for translation. All right, so this is a visual representation of translation. So translation is happening with the help of the ribosome. In the previous step, you made a transcript, which is the messenger RNA. So how do you know which amino acid is going to be left there? Because there's 64 codons, there are 64 transfer RNAs. And the only way to think of it is kind of a chemical trial and error. And they'll come in, and if that transfer RNA has the complementary base pair that's to the anticodon, so this is the codon that's on RNA, and this is the anticodon that's on the transfer RNA, if it's like, oh, this matches, I'm going to leave that amino acid, and I'm going to exit, the ribosome is going to form a bond between those two, and now it's going to move over to the next three, and then that one's going to come in, and if it recognizes that codon there, it'll leave its amino acid, ribosome will make the bond, it just moves over to the next three, and it'll just keep doing that until it gets to the stop. So that's how that recognition happens. And again, notice that there aren't any T's. This is the part that confuses people, is that if there's A's on the RNA at this point, the complement to that would be U's, because you have two RNAs that are trying to recognize each other so that the appropriate amino acid can get left there. And if you look at, if you looked in the genetic table, you would see that AAA is lysine. And so that's the transfer RNA that has it. So then people ask, like, well, what happens after it's empty? Like, where does it go? And it'll just get reloaded with an amino acid. So there are enzymes that will put the amino acid back on it so that it'll be ready to participate in that reaction again. So I guess translation is probably the, more complicated part, but it's not really any more complicated to me than transcription is. So the end result is that you have a peptide, a polypeptide, which is the beginnings of a protein, of a functional protein. All right, so central dogma said we go from DNA to RNA to protein. So we said that it's a 4-letter code, right? So there's 4 letters on DNA, which is A, T, C, and G. If you're going to make an RNA, you just do complementary base pairing, but instead of using T's, you're going to use U's instead. So notice that when there's an A on DNA, if you're going to make an RNA from that, you have to put a U instead. And so every 3 letters on that RNA came from 3 letters on DNA. And so then every 3 letters on RNA is called a codon, and every codon is equal to 1 amino acid in the genetic code table. So the ribosome goes through every 3 spaces essentially, and it'll hold the messenger RNA there, and then transfer RNAs will come by, and if they recognize the codon with their anticodon, then they'll drop that amino acid and leave it there. And it is the ribosome that makes these bonds in between. And the bonds between 2 amino acids are called peptide bonds. So that's why it's called a polypeptide. So the polypeptide, like the sequence of amino acids, is the primary structure of a protein. Other things will happen to the protein later, but that's just where we stop. We stop at the primary sequence, and we don't worry about the post-translational modifications that happen, which would be a little beyond what we need to know. Okay, so central dogma says you go from DNA to RNA to protein. When you go from DNA to RNA, that's called transcription. When you go from RNA to protein, that's called translation. Okay, so I mentioned that sometimes you can have mutations, and mutations are basically a permanent change in the DNA, in the original DNA. So anything could cause that, I guess, like chemicals, radiation, genes like changing, aging like can cause you to get more mutations. So basically, the DNA polymerase usually does a good job of proofreading, like it'll remove the ones that aren't supposed to be there. But occasionally the wrong letter gets put there. Like you either put the wrong letter, or you could remove a letter, and then that could cause everything to get shifted over by 1. And so anytime the original DNA, if 1 letter gets changed, that's called a point mutation. And there are 4 point mutations that we're going to go over. So they'll also call them a single base pair substitution. Because remember, a base pair is anytime there's an A with a T and a C with a G. So if I change a letter from a T to an A, I'm not going to pair that A with an A anymore. I have to pair it now with a T. So you can see how the effect is on the other side of the strand too. It's going to change that spot. Okay, that spot is going to be permanently altered. So we're going to go through the types of mutations. All right, so this is showing an example where sickle cell is due to a single point mutation where the codon was changed in the hemoglobin gene from GAG to GTG. And that turned the amino acid from glutamic acid into valine. And apparently valine is basically changes the chemistry of the hemoglobin so that you end up getting these sickle cell shape. And then those get stuck in the small blood vessels and causes all the problems associated with sickle cell. So sickle cell anemia, which can be a very serious condition, is literally one letter change in the gene that encodes for normal hemoglobin. And so if that gets altered in this case, where the glutamic acid got changed into a valine, that was due to one letter that changed in the original DNA. And I think with sickle cell, I can't remember, I think you have to inherit like both the genes to actually have full sickle cell. You'd have to have one from each parent. And then because of where it originated from, it's more common in African American people for them to have sickle cell trait or sickle cell. All right, this is showing normal DNA sequence. Okay, so nothing has happened to the sequence. So you have your original DNA, and that's how you make your RNA by complementary base pairing. So if there was a T on the original DNA, you got to put an A on RNA. But if there was an A, there's no T's in RNA, so that's where the U comes instead. And then, with the help of the ribosomes, you would translate that using the genetic code table. So every 3 letters is going to mean an amino acid. Okay, and all the amino acids have a 3-letter abbreviation that you would put. Okay. Now this is showing what's called a missense mutation. And that's what led to the sickle cell trait. A missense mutation is where one letter actually changes the amino acid. So the original DNA, there was a change. Let's see if I can figure out where they put the change. The original sequence was that last amino acid was supposed to be a glycine. And then they've altered it in dark red there. So they changed it to a T, which changed that into an A on the other side, and then it made serine instead of glycine. And we'll see an example of this again. So we changed the amino acid. That's called a missense mutation. If it actually changes the amino acid, that's called a missense mutation. Okay, and that's what happened here. It went from glycine to serine. And so those are 2 different amino acids. Okay, nonsense is where you make an early stop codon. You've turned it into nonsense. You've stopped it early, which means the protein is going to be what's called truncated, which means shortened. And a short protein is probably not going to work because it's literally not long enough to do whatever it was going to do. So when you have an early stop codon, that's called nonsense mutation. So you've interrupted the sequence and put an early stop instead, and then that means that the protein is going to be short and it's not going to work properly. Okay, the next one that's the worst, I guess, of them is the frame shift mutation. So this is where you delete a letter accidentally or insert an extra letter. Because what that's going to do is shift everything over by 1. So if you remove that A, instead of reading A, now you're reading AAC. And then you're going to read all the other ones by 1 letter over. And that's why that's called a frame shift. You've shifted the reading frame for the ribosome and the enzymes that are making the RNA. So RNA polymerase is also going to get shifted over by 1. And then that means that potentially every amino acid in that sequence is going to be wrong. And that's why that one is particularly troublesome if you do that. Because if you delete a letter, then you're off by 1 everywhere after that. So imagine when you lose your place in a book and you accidentally read something that you weren't supposed to read yet, like you kind of spoil it for yourself. I think of it the same way, like you lost your place by 1. And so everything is shifted over by 1. And that's called a frame shift mutation. Okay. All right, so let's go to our practice sheets. We'll start with this one. It's double-sided. lesson review. So first of all, if we look at the picture and what's happening, is that a prokaryotic or a eukaryotic organism? It's eukaryotic, right? Because it's got a nucleus. So this is showing an example of a eukaryotic cell, what's going on. So they're showing the double-stranded DNA is inside of the nucleus. And then it's going to get transcribed into messenger RNA, and then that messenger RNA is going to get taken by the ribosome, and then it's going to make the amino acid sequence using the transfer RNA. The transfer RNAs are the ones that are going to bring over the correct sequence. All right, so number one, DNA molecule is blank-stranded, and RNA is what? Double and single. You'll also see double-stranded abbreviated DS sometimes. And then single-stranded is RNA. And you can see this on the picture, too. Like, you can see that the DNA was double and then the RNA is single. Okay, DNA has what sugar? And RNA has what sugar? Why is it called DNA? Deoxyribose. And then RNA has ribose. You're going to be asked to list the differences between DNA and RNA. There's like three main differences, right? One's double, one's single-stranded. One has deoxyribose, the other has ribose. And then one has A, T, C, and G, and the other one has U, C, G, and A. So the bases in DNA are A, T, C, and G. That's adenine, thymine, guanine, cytosine, or cytosine, guanine. And then on RNA, it's A, U, C, G. So you don't have any thymines on your RNA. DNA replication is a synthesis of how many exact copies of genetic material? What happens after you synthesize your DNA? How many DNA do you have? Two. Okay, you go from 1 to 2. Then the process by which a strand of RNA is copied from the DNA. So said another way, what's it called when you go from DNA to RNA? Transcription. You're making a transcript of the original message. And then 3 letters on DNA is called a triplet. And then 3 letters on RNA is called what? A codon. Every 3 letters on DNA will encode for how many amino acids? 1. So it's a 3 to 1. 3 letters on DNA is going to equal 1 amino acid. And that's 3 letters on RNA, which is called a codon. Okay. DNA replication and transcription both occur in what organelle if you're eukaryotic? Nucleus. And I'm going to put a note if you're a eukaryotic organism. Okay, and then what's the site of protein synthesis? The ribosome. Okay, the ribosome. And you remember like when we talked about prokaryotic versus eukaryotic, and we said that basically the bottom line was that all cells have ribosomes. It doesn't matter what kind of cell you are because it has a critical function for life, which is to make proteins. Okay. The process of protein synthesis is also known as the other T word, translation. So the ribosome, with the help of its other RNA buddies, is going to translate the transcript into now an amino acid sequence. Okay, blank is the molecule that brings the correct amino acid for the newly synthesized protein. Which RNA was that? T. Messenger. Yeah, so messenger is the original message that the ribosome grabs onto. And then transfer RNA transfers the correct amino acid over. So that's how I remember what it does. tRNA stands for transfer RNA. It transfers over the correct amino acid during translation. The molecule that helps the ribosome come together for protein synthesis is ribosomal RNA. Like it's literally attached to the ribosome, I guess. So that's why it's called rRNA. So yeah, it gets a little confusing because there's 3 different kinds of RNA that we talked about. Messenger RNA is what was transcribed from the DNA. And then ribosomal RNA is literally attached to the ribosome. It helps during translation. And then transfer RNA transfers in those correct amino acids. So over here, Jesse, this guy here, that's the transfer RNA. And so it's got this little sequence on the top here that's called an anticodon. And it's going to recognize the codon. These are codons that are on the mRNA. So I think when I was in school, I know that was one of the things I was confused about. I was like, why are there so many RNAs? And then I just had to accept that there just is. And so they just do different things, I guess. Okay, proteins are strings of what linked together by peptide bonds? So what are proteins made out of? Amino acids. So there's different ways you could say this. I could say proteins are strings of amino acids, or I could say amino acids are the building blocks of proteins. So I'm saying the same thing, just different ways. If someone asks you what's a protein made out of, you'd say amino acids. Okay, amino acids that are linked by peptide bonds. And sometimes you'll see, if I'm feeling lazy, I'll abbreviate amino acid AA. Okay. All right. Protein synthesis happens in what part of the cell? The cytoplasm. And that's for both cell types. And if you're like, what's a cytoplasm? It's the inside of the cell. Like it's the inside of the cell that's not enclosed in a compartment. So it's like, think of it as the open room, right? And then what if my little area back here was the nucleus? So this would be the nucleus, and then the rest of that would be the cytoplasm. The sequence of DNA that encodes for a protein. So not all DNA means anything. What are the bits that encode for protein called? Starts with a G. How things are transferred from one person to another. Yeah, genetic. Like, how'd you get your beautiful hair? Genes. How'd you get your pretty eyes? Genes. Okay, so genes. The sequence of DNA that encodes for a protein is called a gene. Said another way, genes are made out of DNA. Okay, like so it's kind of knowing that vernacular, like what the different parts are. All right, on the back, this is going to seem redundant, but it's just more of the same, trying to like beat this into our heads a little bit. So if you will see like a DNA and RNA molecule, and I just kind of ask you like, hey, which one is which, and can you tell me the difference between them? So DNA is double-stranded, and RNA is single-stranded. And then the individual unit, they're both called nucleotides. They're just different kinds of nucleotides. One is a DNA nucleotide, and one is an RNA nucleotide. So that's these guys here. Those are the nucleotides. That's what the DNA is made out of. And then it's attached to a base, and that's where the ATCG comes in. So then we asked about the sugar, what the difference is, like why one's called D and one's called R. If you look right here on each of the sugars, notice that the deoxyribose is missing an oxygen. It's just that one little difference right there. The sugars are like pretty much the same. They're 5-carbon sugars. But do you guys see how up here it has an OH and down here it just has an H? The oxygen is not in that sugar. That's why it's called deoxyribose. It just has one oxygen missing. So the sugar in DNA is called deoxyribose, which means that oxygen is gone. And then in RNA, it's just called ribose. So they're both 5-carbon sugars, but one is missing that oxygen, and that's where the deoxy comes from. Okay, the structure, we said DNA was double-stranded helix. Double-stranded helix. And then I'm going to say this is single-stranded, so it's just linear. It's not like curved or anything. Okay, then what are the bases that are in DNA? What are the base pairs that we said, the letters? Yeah, A, T, C, and G. And then on RNA, U, C, and G. Okay, usual location in the cell. So if you're eukaryotic, where's your DNA? It's in the nucleus. And if you're prokaryotic, it's in the nucleoid, actually, which is the region in the cytoplasm where it's like this irregular space. Like, I imagine, like, I don't know, if you threw a pair of shoes of yours in the same spot, relative, like, all the time, like, you're just like, I just throw them in that corner, and that's kind of, but it's like, anyone could go in that corner, right? Like, someone could move your shoes, I guess. But anyway, bacteria are like, let's just put it over here, like, in this space, okay? We don't have, like, a compartment for it. And then RNA, if you're eukaryotic, it's in the nucleus and the cytoplasm because it'll leave the nucleus. So it's nucleus and cytoplasm. And then again with bacteria, it's just in the cytoplasm because they do everything out in the open in one step. Transcription and translation happens in one step. Okay, so the function of DNA, what is it? It's your genes used to transmit traits. And then the function of RNA, it depends on what kind of RNA you're talking about, right? Because there's 3 different kinds. Messenger RNA is, I guess I would think of it as a template for translation. And then transfer RNA, as we said, it transfers the amino acids over. And then ribosomal RNA helps the ribosome translate. It helps stabilize that messenger RNA. So the RNA, the message that's in RNA is the same that was in the DNA. So your DNA is made out of genes, but your body's like, the ribosomes don't read DNA, they have to read RNA instead. So that's why you got to transcribe it first. So messenger RNA is a template for translation. Transfer RNA is going to transfer over the right amino acids, and then ribosomal RNA helps the ribosome, helps it translate. All right, the next sheet talks a little bit about the genetic code again. What was that?
I wasn't done that.
There we go. And I'm going to put up the answer key too. So if you miss some of it, you'll get it. Okay, this is a genetic code. You don't memorize this, you would be given this. I think there's a problem in the practice. If you haven't done the practice quiz, you should do the practice quiz. Like, because it'll show you what I mean. Like, one of the things that you're asked to do is, like, you're given a DNA sequence, and you have to make the RNA sequence. And then you have to make the amino acid sequence, and then you might be asked a question about mutation. So I say, I changed this letter to a T. What happened? Like, what was the outcome? Okay. All right, so here's the genetic code. Remember, there's 64 codons in there, but there's only 20 amino acids. Some of them mean stop, which just means literally stop. That's the end of the protein. And then there's a universal start codon, which is this AUG right there. So 3 nucleotides on DNA are transcribed into 3 corresponding nucleotides on RNA. 3 nucleotides on RNA are called a codon because each codon encodes for 1 amino acid. With the help of the ribosome, transfer RNA molecules will recognize the codon on the messenger RNA and transfer the correct amino acid during translation. Since there are 64 codons to recognize, there are 64 transfer RNA. Only the ones that recognize the codon will leave their amino acid at the ribosome synthesis site. So that's how you know which one will need to be left by reading the code, and then only the ones that recognize the code will leave their amino acid behind. All right, so if you're given this DNA sequence, and actually, I always forget that this one has this should be a TAC to start with. And you'll see why, because I need to have the universal start on there. All right, so if that is TAC, then what's the complementary DNA sequence going to be? So just DNA though. A, G, because I wanted to move that to a C. That's what I was saying. The first one needs to be a C. So I'm going to say ATG. And then what's the next three? ATT, because I asked what was my complementary DNA, right? And then the next one, CAA. And then the next one, the last one, CGC. So it's like inversed, right? That's the complementary DNA strand. So sequences are going to be typically longer than that in real life. They're going to be like hundreds of them. But obviously, I mean, it's redundant. Like, I'm not going to ask you to do hundreds. I think I usually ask you when it's like 6 or 7 long. And so that's the complement. All I did was anywhere I saw an A, I put a T. Anywhere I saw a C, I put a G, and vice versa. That's how you make the complement DNA strand. Because remember, it's double-stranded. So it has to have another side to it. All right, now how are we going to make the RNA sequence? You could do that, and that's super smart to notice. But I usually make people just use the original strand because they get confused by that. But that is correct. Literally, all you would have to do is take the complement strand and add U's where all those T's are. If your mind works like that, great. But if not, then you just use the original strand to do it, and you'll get the same answer. So that's a really good point. It just depends on how your brain wants to work it. So if you understand that, then you could do that. Or you could just look at the original strand, and I'm going to add complementary base pairs, but I'm making an RNA now, so I can't have any T's. Okay? So when I see an A or T, I can still put an A, right? Because I can still put A's on my RNA. But the switch comes is that when I see an A, I'm not going to put a T anymore. I got to put a U now. Because now I'm starting to make it into my genetic code. And then I have a C, and I'm going to put a G. Does everyone see what I did there just initially? Since I am now making an RNA, I cannot use T's anymore. So I'm just going to make complement base pairing using U's instead. So U is taking the place of T. And so if there was a place where you were going to put a T originally, you got to put a U now. Okay? All right, so we got AUG. And then what comes next for TAA if I'm making an RNA? AUU. Right? Because I can't put any T's there, but I can put U's. And then the next one is still going to be CAA. And then the next one is still going to be CGC. So what Jesse was saying is that what you could do, which maybe would be easier, is if you already made the complementary DNA strand, just put U's in it instead of T's, and you'll get the exact same answer that I just got. Does everyone see how that would be equivalent? Like if you just use them as the same? Okay. So anytime you're asked to do any kind of manipulation, like if you're asked to make an RNA or you're asked to make a mutation, you need to use the original strand that you're given. That's the safe thing to make sure that you just get the right answer. So you always refer to the original strand, which is called the template strand. It's what's going to be a template for everything else after that. All right, now that we have an RNA sequence, we can go to the table and find the amino acids. So what does AUG encode for in the table? MET. Yep, MET. And then what's AUU? ILE. Sorry, the font is off. It's ILE. And then what's CAA in the table? Yep, it's GLN. Yeah, sorry, the font's kind of weird. And then what's CGC? ARG. ARG. Okay? So that would be the amino acid sequence. Now, if I was asking you on a test, of course I'm going to put a stop. And that's how you would know that you got the right answer, probably, because you have a stop there. So you should start with methionine, and you should end with a stop if you did it the right way. All right, now in the second scenario, I'm asking you to go backwards. So I already gave you the RNA. I'm asking you to go back and tell me what the DNA was. Okay? So if my RNA says GCU, what should my DNA have been? C, G, A. Sorry, I typoed here. C, G, A. Okay, does everyone see that? Because this came from the original DNA. So it has to complementary base pair with the original DNA. So if this is a G, that means the original DNA should have been a C. And then this is a C, it should have been a G. And then this is a U, so that means that it must have been an A. Because we don't have T's on RNA, but we have a U there, and had to appear with an A. So that's how I got the first one. I wouldn't ask you, this is harder than I would ask you to do. But if you can go backwards, then that means you definitely can go forwards. Okay, then what would AAA be on DNA? TTT. Because you can have T's on your original DNA. We're going backwards now. And then what would CAA be? GTT. And then what would GGG be? CCC. And again, I'll put up the answer key to this. All right, now going back like forward then, what would the amino acids be using that RNA that we're given? ALA, A-L-A, okay? And then what's AAA? Lysine, L-Y-S, okay? And then what's CAA? GLN. And then GGG? GLY. Okay. All right, the last scenario, we are starting with the amino acid sequence. And this one is probably, it's not really tricky. You'll just see that there's more than one answer possible. And that's what we meant about the code being degenerate. So how many amino acids are there for phenylalanine, PHE? There's 2. What were the codes? And UUC? Okay, those could have been phenylalanine, right? And then what's the only code for methionine? AUG. And then what's the codes for histidine? HIS. CAU. And anything else? CAC? Is there anything besides that? Histidine, I don't think it pops up anywhere else. And then how many codes are there for serine? There's 6 codes for serine, right? I'm not going to write them all out. But if you went and found them in the thing, you'd see that there were 6 of them at least. Okay? And then if you went backwards, I'm not going to do that right now because it's kind of like redundant, but you would do the same thing where like, if this was UUU, then it should have been AAA on the original DNA. So the RNA has to come from the DNA. So any changes you make would be in the original DNA. All right, so let's do, I'm going to try to make up one in my head. This never goes well. I don't know why I didn't just bring my example. All right, I'm going to start making up a DNA strand. Okay, so let's say I have TAC, TAT, CGA, TGC, and ATT. Okay, there's my DNA strand. So what's my complement DNA? Different colors here. Okay, ATG, ATA. I already have a C there.
GC.
Uh-huh. And then, oh, sorry, yeah. I've all stripped it over. ACG, and then TAA. Okay, so we're kind of drawing it like in a line. But imagine if I drew it like this, and this was one strand, and I had T, A, C, T, A, T, C, G, A, for example. The other strand is A, T, G. Like that's what you guys are doing right now. Did you see that? I don't want to do the whole thing, but hopefully you can see that that's the pairs that you're making. So this is the template. It's basically whatever I give you to start with, that's what you should like refer back to, like if you're asked a question. And then this is the complementary. And we know that we can make the complementary because A always goes with T and C always goes with G, and it doesn't matter what side of the strand, like it's always going to be complementary. All right, so then if you were asked to make the RNA, the messenger RNA, you're going to use the original strand to do that. Otherwise, you're going to get some weird backwards thing. And so if we see a T, then what is the complement to that? To A. And then if we see an A, it's U. Because we don't have any T's on our RNA anymore. We're just making a one-letter switch. And then C, we're going to put a G. And then we're going to say A, U, A. And then we're going to say what? Uh-huh. Uh-huh. And then U, A, A. And what Jesse was saying was that you could also take the complement strand and replace all those T's with U's, and you'll get the same answer that we just got, which is a nice little trick. But it's whatever your brain can handle, okay? All right, now we need to know the amino acid sequence. So where do we find that? We just make it up? No. Yeah, we just look in the chart. So can you guys tell me what AUG is?
MET.
MET. And then what's AUA?
MET.
No, it should be something different.
Oh, ILE, yeah.
ILE. Okay. And then what's GCU?
ALA.
ALA. And then ACG?
Stop.
ACG shouldn't be a stop. The
Oh.
last one is stop. UAA.
GC.
Wait, what? THR, right? Threonine? Is this one?
Yes.
Okay, do we have all our amino acids? How many amino acids do we have? 1, 2, 3, 4, stop. So if you were asked, how many amino acids are in the sequence, you'd say 4. Because methionine is both a start and it's literally an amino acid. And this came from the genetic code table. So we just looked it up and we got methionine, I always forget what that is, isoleucine, alanine, threonine, stop. Okay, so then the last part, which I guess would be the trickiest part, would be to try to make a mutation of some sort. Now to make a mutation, you have to do it in the original DNA. So let's say that see if I can figure out one. I don't know, I'm just going to randomly do one and see what happens. What if I go back to this original DNA up here in this spot that I put a star by? And what if I change that to a T? Just something happened. I made a mistake. If I change that to a T, then I'm going to I had CGA, right? And now I've mutated it to TGA. Does everyone follow me? I just randomly made a mutation to see what would happen. Now, we said that CGA on RNA is GCU, and I've just changed it to something else, right? I've changed it to ACU. When I look in the table, are those different amino acids? So you have to look in the table. We said GCU was ALA. And then what's ACU? I'm sorry, can you say it again? THR. Are those different? Yes, that's called a missense mutation. So it's not enough for you to just, usually on the question I'll say explain. If you say it's a missense, but you don't show me this, this is literally all you have to show me. You'd say, okay, this was the original DNA. And then we mutated that one letter. That's why it's called a point mutation. We changed it to a T. When I look at the RNA, I realize that my RNA went from this to this, which is going to be different, and I just know that from experience because you change the first letter, it's probably going to change it. And then it did change it, because when I looked in the genetic code table, it went from an alanine to a threonine. And if you change the actual amino acid, that's called a missense. Now, I can't remember off the top of my head, but what if alanine and threonine were, pretty similar, chemically? could the change still maybe be okay? Maybe, right? That's the difference between a conservative and a non-conservative missense mutation. Conservative just means that you did conserve the function, because even though the amino acids changed, they were similar enough that it was still okay. I can't remember off the top of my head if these are similar enough. But sometimes it can be okay. Now, what if I changed it and I did not change the amino acid? That would be called silent mutation. So technically, I changed the letter, but if the amino acid had ended up being the same, then it doesn't matter. Like, you wouldn't even know that that happened. So it would be a silent if that happened. So I will post the practice I have with the answer solution. And then I think on the video I like talk about this, like I do an example or whatever. Okay, all right guys, that's it. So we'll take a little break.